Hello, everyone. My name is Dr. Wlliam Kupsky. I'm a neuropathologist and professor of pathology at the Wayne State University School of Medicine in Detroit. I'm going to talk briefly about the pathologic diagnosis of multiple system atrophy. Multiple system atrophy is an example of a neurodegenerative disease. The general definition of neurodegenerative diseases is an idiopathic, systematic, progressive loss of neurons that results in a gradually progressive loss of neurological function. To break this down, idiopathic means that in general, we don't know what the cause is, although much research is being done to investigate this. I'll define systematic in a minute, but most neurodegenerative diseases are characterized by progressive loss of neurons, the basic functioning cell in the brain, and this results in loss of brain function. Diagnoses of neurodegenerative diseases are made by clinicians evaluating different aspects of neurologic function, such as cognition, motor functions, sensory functions, or affective or motivational functions. Neurodegenerative diseases generally affect one or more of these areas. Multiple system atrophy, for example, primarily affects motor functions. As a pathologist, the basic pathology of neurodegenerative diseases is to look at the brain and evaluate areas where nerve cells or neurons are lost. These are usually accompanied by proliferation of a supporting cell in the brain called a glial cell that produces what we refer to as scarring in the brain or gliosis. In fact, in the neurodegenerative diseases, which parts of the brain are involved, defining a pattern of neuronal cell loss often defines a particular disease, and we'll see that with multiple system atrophy. And then some of the diseases have particular structural changes that are characteristic of that disease, and we'll also see that with multiple system atrophy. The clinical diagnosis of neurodegenerative disease is often made by ancillary clinical tools such as radiological imaging by the neuroradiologist or electrophysiology, EEG, by electrophysiological neurologists. From a pathology point of view, the clinical lab is sometimes used for testing for diseases, looking at blood or cerebrospinal fluid for particular markers. Unfortunately, for many of the diseases, specific markers are not available, and that includes MSA. So we are generally reduced to looking at parts of the brain or the entire brain, tissue sampling. If we look at the whole brain, obviously that has to be done on autopsy or postmortem examination. So we look at the brain or spinal cord, and for some of the diseases, we may investigate other relevant tissues such as peripheral nerves or muscle. When we have tissue available, sometimes it's helpful to do molecular testing on those tissues. The characteristic features then of neurodegenerative diseases that we look for as pathologists are a systemic neural loss in the brain that doesn't correspond to a particular region or vascular territory. And this is a brain system such as the motor system, the visual system, the sensory system, the cognitive system. Generally, diseases involve both sides of the brain, so they're bilaterally symmetrical. Many of the diseases involve only the central nervous system, but some also involve the peripheral nervous system, in which case we would have to examine that as well. And if we lose neurons in the brain, we generally lose brain volume, so the brain becomes atrophic. That can be appreciated by the neuroradiologist as well as by the pathologist. That's due not only to the loss of the neuronal cell body, but also the processes of the cell that we refer to as axons and dendrites. Those are the connections that one neuron makes to another neuron. The loss of neurons is often accompanied by proliferation of cells called glial cells or astrocytes. This leads to what we refer to in the brain as gliosis, which is the brain's method of scarring. This generally occurs in the same areas that the neurons are being lost. As a pathologist, then, we correlate our findings with the clinical findings, and that includes the pattern of brain involvement. Does that match the clinical and the imaging findings with our pathologic findings? We look for these particular histologic inclusions or characteristic features. Sometimes we actually have to count them and quantify them, and then once in a while, with increasing frequency, molecular testing is done to confirm the diagnosis. To turn specifically to multiple system atrophy, this is a sporadic neurodegenerative disease, meaning it occurs randomly in families, usually not inherited. It's characterized by progressive abnormal loss of movements or abnormal movements, and it's accompanied by accumulation of a particular protein, generally occurring in glial cells, but also sometimes neurons. From a clinical point of view, there are usually two major patterns. One is referred to as MSAP, where the patterns resemble Parkinson's disease, and this involves particular structures of the brain referred to as the basal ganglia. This is sometimes referred to as the striatonigral system. That may be the case according to some studies, in about a third of patients with MSA. A second type is called the cerebellar form, or MSAC. This involves particular structures in the brainstem and the cerebellum, another part of the brain, and I'll show a picture of that in a minute. This is an olivopontocerebellar pattern that usually is less than 20%. Interestingly, almost half of the patients with MSA will show involvement of both regions of the brain. The specific feature characteristic of MSA that we look for as pathologists is accumulation of this alpha-synuclein protein in glial and neuronal cells in these affected areas of the brain. To look at the particular structures involved in MSA, after we examine the brain, we look at slices of the brain and then sample it for the particular structures. For the MSAP version, as indicated here, we're looking at structures such as the putamen, which is part of the basal ganglia, the substantia nigra, but also many other structures. For example, the locus ceruleus, which is in the brainstem. In the olivocerebellar portion of the form of the disease, we look at brainstem structures such as the basis pontus, middle cerebellar peduncle, cerebellar cortex, or inferior olivary nucleus. These are all complicated terms, but they refer to specific brain regions. For example, the substantia nigra, the putamen is located in this part of the brain known as the basal ganglia. This is the putamen, the globus pallidus, and the internal capsule. The substantia nigra is located in the midbrain. This is the upper part of the brainstem. That's this dark area right here. For the MSAC form, we would look at structures such as the basis pontus. This is farther down in the brainstem, this area of the pons called the basis pontus, and its connections to the cerebellum. This is the middle cerebellar peduncle. We might also look at the cerebellum. This is the cerebellar cortex, and its white matter underneath the cortex. And then we may look at a caudal part of the brainstem in the medulla at a structure called the inferior olivary nucleus located here. In order to look at the brain, obviously we have to perform a postmortem examination. Postmortem examination requires permission of the family, often with request of the family or request of the clinician prior to obtaining the specimen. Once we remove the brain, we may save parts of it frozen or with special processing, such as fixation in a chemical called glutaraldehyde, to preserve the tissue for other types of tests. The specimen that I just showed is an example of a brain that has been fixed in formaldehyde or formalin. Fixation preserves the brain, changes its color, but it makes it firmer and enables us to slice it, as you saw in the previous slide. What we did in the previous slide was look at the different parts of the brain. That's called the gross examination. We will usually write a report upon that. And then from the particular areas that we were looking at, for example, the putamen or the substantia nigra, we will take samples for microscopic examination. These samples are embedded in paraffin wax to facilitate making very thin slices, and then the slices are mounted on glass slides. These slices are very thin. They generally range from 8 to 10 micrometers in thickness. We will do special stains or studies on them. This is an example of a commonly used stain in autopsy called Luxalfast blue with hematoxin and eosin. The pink is the eosin, the blue is the Luxalfast blue. The blue is staining the white matter, the pink is staining the gray matter in the brain, and this shows you the putamen and the two parts of the globus pallidus that we saw in the previous gross specimen. Now, what are we looking for, particularly in MSA, is aggregates of these alpha-synuclein protein. Interestingly, for a lot of the neurodegenerative diseases, the abnormal protein that accumulates is really a normal protein. Alpha-synuclein is a 140 amino acid protein that's encoded by a specific gene called SNCA that's present on one of our chromosomes, chromosome 4. This protein is normally present in neurons, usually in the nucleus of the cell, that's the central part of the cell, but it's also present in some of the processes and the interconnections between the cells. It's thought to be associated with synaptic activity, but its actual function is incompletely understood. For reasons which are also incompletely understood, the protein is upregulated in patients with MSA and accumulates in different structures. It may, for example, accumulate in the glial cells, the supporting cells of the brain. These are called glial cytoplasmic inclusions, or GCI. It will also accumulate in neurons in involved areas of the brain. These are called neuronal cytoplasmic inclusions, or NCI. We may also have the protein accumulating in the nucleus of these cells, not just the cytoplasm. So those are neuronal or glial internuclear inclusions. And then it may occur in the processes of the cells, the axons or the dendrites, and these are referred to as neuropil threads. For example, if we were to take a sample from the basal ganglia, this is again similar to the slide I showed, where you see the putamen, the globus pallidus, the internal capsule, and the caudate nucleus. This has been cut, embedded in paraffin, cut into a thin slice, and stained with Luxelfast blue PAS to show those structures. And this is what that looks like under the microscope. If we compare the appearance of this to the normal putamen on top here, the normal putamen has all these neurons scattered throughout it in a very finely textured background. This is called the neuropil, and that's the meshwork of wires interconnecting all of the cells. In the lower picture here, all of the neurons are gone, as well as their axons and dendrites, so that's produced a reduction in density of the tissue. And then the small cells here with the small nuclei are the glial cells, or astrocytes. Instead of being small and inconspicuous, they have proliferated. They also make cytoplasmic extensions called processes, and they form a very loose meshwork. So this is the neuronal loss and the gliosis throughout the putamen. It's very hard to see the abnormal inclusions, but if you look very carefully at the glial cell cytoplasm, some of it is a little darker pink, that's normal. Some of it is a little lighter pink. As we'll see, the lighter pink is the abnormal inclusion in the cytoplasm. In order to see this, we need to do some additional stains. The panel on the right shows the same section stained with alpha-synuclein. The positive staining for alpha-synuclein is brown. This is a nucleus of one of the glial cells, and you can see the cytoplasm is now filled with this brown material. That was that small pink aggregate that we saw on the H&E stained section. So this is showing numerous inclusions in the cytoplasm of the glial cells. Those are the GCI, and then the fine lines in the background that are brown represent the processes of these cells. These are examples of neuropil threads. It's sometimes helpful also to highlight some of the other cells. This stain, glial fibrillary protein, or GFAP, highlights the reactive astrocytes. You can see from this stain why they're called astrocytes, which means star-like cells, because of these radiating processes. These are abnormal. They should not be this large or proliferated in the normal putamen. So this is the gliosis. If we were to look at the substantia nigra is a group of cells in the midbrain. Normally, the substantia nigra, when you look at it in a gross slice of the brain like this, is rather dark black. It's dark black because the neurons in the normal substantia nigra produce a pigment called melanin. We see that as brown pigment within the cytoplasm. This is normal. In a patient with MSA who loses neurons in the substantia nigra as well, the substantia nigra loses its gross intensity. It's no longer dark black as it should be, and under the microscope, you can see a reduction in the density of the pigmented neurons. There's still one or two left here, but most of the neurons are gone. The background is density is reduced, and the glial cells are proliferated. So the substantia nigra is depleted of neurons and gliotic, with alpha-synuclein inclusions in some of the remaining neurons. That's confirmed here with the alpha-synuclein immunohistochemical staining. This is a neuron filled with alpha-synuclein in the neuronal cytoplasm. One other structure that we might look at, and normally to make the diagnosis of MSA, we would sample many different structures in the brain and look for the presence or absence of these inclusions and the presence or absence of neuronal loss and gliosis. This is a cerebellum in a patient with MSA. The cerebellar cortex itself, there's a little bit too much space between the folds of the cortex, so it's slightly atrophic. And our section here includes the cortex, a little bit of the white matter underneath, and one of the gray matter structures within the cerebellum called the dentate nucleus. This is the histologic section. This is a sample of normal cerebellar cortex where you can see different densities of neurons, but most of the purple dots are neurons. These large cells are Purkinje cells. Those are one of the major neurons of the cerebellum. If you look at the patient with MSA, these cells are missing. So there is loss of neurons in that population. If we look at the underlying white matter, which is the axons, this is dark blue because of the presence of the myelin around the axons. Underlying the cortex in the patient with MSA, there's a reduction in the density. This is what that looks like under higher magnification, normal white matter, and then MSA, where there's been a loss of the myelinated axons. And again, you see these glial cells in the background, many of which contain the gliocytoplasmic inclusions. This is a higher power view of that to show the reactive astrocytes in the background, some of which have these very small pink clumps in the cytoplasm. They would be highlighted again with the alpha-synuclein immunostaining. So without wanting to show many of the other structures that are involved in the case, the pathologic diagnosis and confirmation of MSA requires examination of postmortem brain tissue. We need to look at the brain in autopsy. Then we need to sample different areas of the brain to determine the pattern of neuronal loss and the pattern of gliosis and look for the alpha-synuclein aggregates. If we find the aggregates and the cell loss primarily in the basal ganglia, substantia nigra and subthalamic nucleus and others, we have a diagnosis of MSA, P, the striatonigral pattern of MSA. If we find them primarily in the basis pontus or pontocerebellar projections, cerebellum or inferior olivary nucleus, we have the MSAC form. In the case I just showed you, all of those sections were from the same patient, so that is a mixed pattern where we have elements of both. The alpha-synuclein aggregates, to summarize, include inclusions in both neurons and glial cells, as well as their cell processes. So pathology is necessary for the confirmation of the diagnosis of MSA, and I hope that this provides you with an understanding of what we are doing as pathologists when we examine the brain in autopsy. I've provided a few textbook references in case you're interested in looking at these. These are detailed medical textbooks, but they will show often very interesting pictures similar to what I projected to you. Thank you for attention, and I hope you enjoy the rest of the conference.